Okay, so hi everybody. Yeah, so this is the last day for revision. So the last chapter for our syllabus lah, one coming out exam. So I'm going to start first. So here we have a summary chart that me and Philbert made. All right, so it covers like summarizes almost the entire thing. Okay, so here the new thing that you have to know is just the 11.2 and some bunch of other subtopics here. So for this summary chart, I'm only showing you the 11.2 part, which is the immune response. So the 11.1 is just basically the introduction to phagocytes. So I'm pretty sure all of you know already, right? So just a short recap. We have the types of white blood cells here. Okay, you can see here all of this. So white blood cells are divided into two parts. Okay, so it's granulocytes and agranulocytes. All right, so they are granules. So the ones that we should know are the phagocytes and lymphocytes. So the ones that we're only going to be learning is neutrophils, monocytes, and lymphocytes. So all of these participate in the immune response. All right, so that's just a brief introduction. So phagocytes, you have neutrophils and monocytes, which will turn into macrophages in the tissue. And then for lymphocytes, which participate in the specific immune response, will be the B lymphocytes and the T lymphocytes. Okay, so moving on to 11.2, this is what you should know. Okay, the definitions and all that stuff. So immunity, you have 3 definitions over here, 1, 2, and 3. And then you should know that immunity is divided into 2 parts, the non-specific and specific. So this chapter is actually quite, we just learned it, right? Quite new lah. So just to refresh and recap everything. Okay, so you should also know that we have 3 lines of defenses. This is the first and second will be non-specific. And then the whole thing here is the 3rd line, which is very specific. Okay, so there's just a brief summary of everything, how they work, the phagocytes, and then the immune responses. So we're divided into 2, the cell-mediated and the humoral one. Okay, so you should also know the term antigen here. We got 2, non-self and self-antigen, and what is also antibody. Okay, so at the side here, these in the specific response, we have the primary phase. Okay, it just basically involves the whole thing. Then the secondary phase is where memory cells are made. So memory cells here, they're very important, okay? So this is a short summary. Okay, so I'll also be looking at the antibodies here. So we just have a diagram here for antibodies, okay? So here, the variable region, okay? And down here will all be the constant region. Okay, so this one binds to antigens and they're made out of different polypeptide chains. So down here will just be the same, everything's the same for all the antibodies. Okay, so yes, you can go through a bit, okay, and how they work, okay, all the hinge region, all that stuff, okay. So, and then also we have the mechanism of how these antibodies work. So we got 1, 2, 3, 4, 5, 6. So very easy to remember. Only the names only a bit like bit tongue twisting lah. Okay. So but either way, easy way to remember lah. I just remember like this. So for the mechanism of the antibodies. So I just do, oops, sorry, it's too thick. Don't like that. Okay. So I just remember like this. No, meal. This is how I remember, like, you can just choose whatever way you want. So N stands for neutralization, N stands for neutralization, then O, opsonization, M is to prevent. So M and E, M stands for movement, and E stands for entry. So they both of them prevent movement and entry. So and then A is agglutination and L will be lysis. All right, lysis meaning breakdown. Okay, so I'm just gonna cut this short, yeah. Okay, so the next part, Philbert is gonna handle them. All right, Philbert. So
okay, so let's start off with the monoclonal antibodies. So definition is the immunoglobulins or glycoproteins produced by a single clone of plasma cells. The production of monoclonal antibodies is actually through the hybridoma method. Basically, you inject a specific antigen into the mouse to stimulate an immune response to produce antibody. And then the mouse is killed, and then the spleen is removed and then flushed to remove the plasma cells from the spleen. The plasma cells then fuses with the cancer cells to produce a hybrid cells called hybridomas. The hybridomas is then cultured to undergo mitotic division to produce a clone of hybridoma cells. The hybridoma cells will then be added into this microtither plate. And then lastly, they will be screened for production of the desired antibody. So yeah, so the uses for monoclonal antibodies mainly is to diagnose and treat diseases. You can see here to diagnose, you can determine blood type, you can determine the tissue type, locate the blood clot, tumor, and then you can identify pathogens such as HIV. You can distinguish different strains of pathogens. You also can distinguish different types of leukemia, and lastly, pregnancy tests. For treatment, you can treat breast cancer using the trastuzumab, and then treat skin cancer known as melanoma using the ipilimumab. You also can treat the rheumatoid arthritis, or also known as RA, using the infliximab. And lastly, you also can treat leukemia and autoimmune diseases using the rituximab. Next, we're gonna talk about the role of memory cells in the secondary immune response as well as the long-term immunity. So during the first antigen injection, the antigen enters the body for the first time. So that's why a primary immune response is triggered. The primary immune response is triggered, so there will be a slow antibody production, resulting in less antibody produced. So because of this, the pathogen may be able to spread in the body, causing the infection to develop symptoms. The reason why there's a slow antibody production because the T helper cells or the T lymphocytes need to undergo clonal selection and expansion to actually produce the memory T helper cells and the memory T killer cells. Also, because there are less B cells to start with, the B cells also need to undergo clonal selection and expansion to produce the memory cells and plasma cells. Only then the plasma cells can produce the antibodies. So the concentration of antibodies will decrease because the plasma cells will die, and also the antibodies that are binded to antigens will be phagocytosed. So during the second antigen injection, a secondary immune response will be triggered. The antibody production will be rapid and more antibody produced. Because of this, the pathogen can be destroyed completely before any symptoms is developed. So this causes infection without symptoms, reservoir, and transmission. The antibody concentration also lasts longer. This is because the secondary response actually bypassed the clonal selection and expansion, because the T helper cells and as well as the memory cells are already produced during the primary response. So next we're going to talk about the types of immunity. The types of immunity can be separated into active, which is when the own immune system is activated, usually is when an antigen is encountered, and passive, where the own immune system is not activated, but an external antibody is introduced. The active and passive immunity can be further divided into natural and artificial. Natural is acquired through normal life processes or another organism. For example, in this case, active natural immunity would be breastfeeding, or either through across the placenta. And then artificial would be acquired artificially, so deliberately by immunization. Mainly is vaccination, and then For passive artificial, it's serum, which is basically an injection of antibodies. And passive natural would be, oh, sorry. Oh, yeah, sorry. Active natural is not breastfeeding. Passive natural is breastfeeding and across the placenta, but active natural is by infection. Yeah, sorry. Yeah. Next is vaccination. Sorry, vaccination. Starting off with the definition below there. Yeah. The foreign antigens is introduced in the body artificially to activate a primary immune response. And in case there's a subsequent exposure to the same antigen, it can activate a secondary immune response to produce antibodies rapidly. So the vaccine, there are a few types of vaccine. You got whole life microorganisms, acute microorganism, attenuated microorganisms, which is basically harmless, a toxoid, which is a toxin, but it's also harmless, and lastly, genetically engineered vaccines. And then you also have the problems with vaccines. Problems with vaccines include all these you can read only for infectious diseases because only cells have antigens, and then live vaccine is more effective, but it can multiply in the body by mitotic division. Poor response due to malnutrition or poor diet, antigenic variation because of many strains, antigenic shift, and also eukaryotic antigen can change in different stages, antigenic concealment, and also there are side effects for vaccines. So how to make vaccination successful mainly is herd immunity and also continuous vaccination. Implementation of vaccination include vaccination programs, national policies by making vaccination for deadly diseases legally required. And lastly, international cooperation like the WHO, cooperation between developing and developed countries. So that's all for the summary chart. Now we'll pass to Sharmaine for the questions.
Yeah, so since we got limited time, okay, we're just gonna start with the questions, yeah. Okay, so first up, it is to recognize your immune system. So yes, this is just the classifications of all the white blood cells that we have in the body. So I'm wondering, does anybody really want to try? I mean, here they asked to, this is a list of some features in cells which involve immunity. So fill in the appropriate boxes by tables by selecting the cells which matches the features from the list below. Okay, so here it also says that some boxes may contain more than one relevant cell. So here it can be either cells or cells, so you can have more than one. So we just go through this, okay, and then the selection will be down here, okay. So the first step, so in order to identify the cell, we have cells that phagocytose bacteria in tissues. The option is down here, yeah. So does anybody want to try? Does anybody want to try the answer? What is the cell?
I think you can call the name. If they don't
Neutrophils. Don't worry, it's Friday. Everybody's in good mood today. Okay, yes, so correct. Today, sorry, it is neutrophils, yeah? Wait, who got it? Who got it? Okay, Katie got it. Tissue macrophages, yes, Katie and Shumin. So it is neutrophils and macrophages, or you can also write monocytes because monocytes are the macrophages that are not in the tissue yet. So the second one will be secret antiviral antibodies. I think quite easy lah. So plasma cells, okay, thank you. Plasma cells, everybody know already. Oopsies, okay, never mind. So release histamine and serotonin allergies. Okay, so this one is not in the syllabus, but still I think it's good to know, like, I mean, it's already there. So it will be use basophils and mast cells. Actually, mast cells are the ones that secrete histamine, right? So they're the ones who call the macrophages to come to the infected site. Okay, so basophils also do the same thing. All right. So next question, manufactured in the bone marrow. Eosinophils. Okay. We should need the answer. The selection is down here, but it's actually correct. Yeah, selections are down here. Monocytes. Okay, some more. Some more. Okay. Actually, all your answers are correct, lah. Because like all white blood cells are made in the bone marrow actually. So yes, all of them. So you can just choose any three, okay? Or T lymphocytes are made in the but then they don't write here, but they mature in the thymus. But still, they're all made in the red bone marrow. Okay, so initially stored in the thymus glands. Yes, correct, Joyce. T lymphocytes. So T lymphocytes. Okay. So main cell humoral immunity. Anybody?
Cell of human. Ayo.
Okay, thank you, Inteng. Yes. B lymphocytes or plasma cells. Okay. So this one's also not in the syllabus, combat effects of histamine. This is actually eosinophils. But teacher, can I just ask lah? I thought eosinophils they promote inflammation instead. So why don't they combat the effect of histamine?
This one, right, is not in syllabus, so we skip. Okay.
Okay. Okay, sorry. Okay, next one, act as phagocyte after transformation in tissue macrophages. Quite obvious. Monocytes, okay. And phagocytosis, the debris of antibody antigen reactions. This one is also not in syllabus, eosinophils. So next one, differentiate to plasma cells, B lymphocytes, yes, B lymphocytes. All right, yes, Ian. Okay, so then differentiate to cytotoxic killer cells is T lymphocytes, then navels. Next one, naval rapid immune response. This one shall be memory cells, yeah. Okay, because it's already been through this before. Okay, so once it sees the same antigen again or pathogen, then it will elicit a quicker response. Okay, so this is what helps it. Then last one, manufactured in lymphatic tissue is actually, yeah, all this. Okay, so the main ones are, so you can just choose any three. All right, so okay, next question.
I think you all didn't learn about lymphatic tissue because this is in the old syllabus.
Yeah,
lymphatic
because
tissue is where the B cells and T cells, that's why lymphatic means it's related to lymphocytes. So that's the location where lymphocytes mature and circulate. Yeah, okay. So basically are the lymph nodes area and the lymphatic vessels. Okay,
so the diagram shows an immune response by B lymphocytes, okay? So here we have a B lymphocyte, okay, B lymphocyte in the lymph. Oh yeah, I forgot to mention all these cells, all these lymphocytes are actually mainly found in the lymph, yeah. So that's why they're in the lymphatic tissue. Okay, so and then after that, there is a helper T cell over here, okay? So what you can see they're doing is that they're exchanging information, okay? Why? Because here there is an HLA antigen. So HLA antigen is self-antigen. Okay, and then this T lymphocyte has a receptor for that self-antigen. So you can see that they're binding to each other, which means they're like kind of communicating. Okay, so why are they communicating? Because you can see that this B cells has brought a processed foreign antigen. So when he brings this foreign antigen, of course, he has to give it to the T lymphocytes. Okay, because they have to do something about it. So then this T lymphocyte also has a foreign antigen receptor for this. Okay, so then the immune response starts to happen. Okay, so just to recap with immune response, okay, so down here, okay. I guess this one we can compare, this one can like explain better because this is for another question. But this happens first, all right? This actually happens first before the immune response happens because this is non-specific. So it's non-specific first and then only specific, okay? But since we just went to this question, so I'm just gonna explain what happens after the phagocyte takes action, Okay, so we just assume that the phagocytes already like they already eat the now I just explain this one. Okay. Okay, so first when the phagocyte sees a antigen, okay, so yes, the mast cells will release histamine, remember? So mast cells are the ones that are the cells that are near the endothelial tissues, for example, near the near the blood vessels, okay? So first, they will release histamine. So histamine does a lot of things, which mainly does promote inflammation and vasodilation. Okay, so this histamine is actually a chemoattractant. So a chemoattractant means they attract phagocytes. So the attraction of phagocytes to the infected site is called chemotaxis. Okay, so number one, phagocytes move towards the chemicals like the bacterial components. The bacteria also releases some chemicals that actually provoke the phagocyte to come to it. Okay, and then they also respond to cytokines and histamine released by mast cells. So yes, this is a neutrophil. Okay, so neutrophil with the granulated, multi-lobed nucleus. Okay, approaching a bacteria. Okay, second will be the recognition and attachment. Okay, so the neutrophil will then attach to the bacterium. Okay, with its specific antibody for the antigen to the antigen of the bacterium. Okay, so then the third process will be endocytosis or phagocytosis. Okay, so yes, they extend the pseudopodia, okay, so to engulf the bacteria and then it will fuse back again. Okay, so fourth one will be digestion, where the lysosome or the primary lysosome will digest the bacteria in a phagocytic vacuole. Okay, so then they fuse, all right, become secondary lysosome or phagolysosome, and then in the end they just digest. Okay, so this is actually what pus is, the yellow thing you see. It's actually dead white blood cells and dead bacteria. Yeah, this is just like a randomness of a bacterial, like a what do you call that, a white blood cell and bacteria fighting like that. Okay, so yes, the bacteria is killed by H2O2. This is definitely toxic, a toxic byproduct of cell metabolism and toxic free radicals. So then bacteria is digested by enzymes. Okay, then the you can say the polypeptide bond is broken up by hydrolysis. Okay, so this is what happens firstly in non-specific. Okay, so once this happens, actually, right, the macrophages, they will become antigen presenting cells. So what do I mean by this? And once they finish digesting the bacteria, they still leave some protein remnants of the bacteria. So what does this macrophage do with the protein? Is that they take the protein, then they present it on top of their cell surface membrane. So it's kind of like the show off lah, but the show off in a good way, because this is very important. So immune response begins with antigen presenting. Okay, antigen presenting. So one of the cells that do this will be the macrophages. Okay, actually, the main ones who actually do this are called dendritic cells, but then they're not on the syllabus. They're considered the professional antigen presenting cells because that's what they do. Macrophages, they do that, but also they mainly eat other cells. Okay, so first slide. Am I going too fast? I'm sorry if I'm going too fast. I haven't time. Okay, never mind. Yes, sorry. Okay, so first, what will happen is that antigen presenting. Yes, antigen presenting happens. Okay, then this by the macrophage, then the B lymphocytes and T lymphocytes will be attracted to this. They will be attracted to the macrophages. All right. The macrophages already go inside the lymph. They went inside the lymph because that's where B and T lymphocytes are mainly found. Okay, so then the B lymphocytes will bind to the antigen on top of the macrophages, the APC. Then also the T lymphocytes also will bind there. So 1 and 2 is antigen presentation. So then next what happens is that clonal selection, all right? So you can see, right, just like we learned before, just a recap, you have different kinds of antigens. So they'll choose the antibodies, they'll choose the specific antibodies, all right, to bind with them, okay? Same goes with the T lymphocytes, right? Then they will select which one has the specific antibody for the antigen. Okay, so then they will divide by mitosis. Okay, he will produce, the T lymphocytes will produce, it will divide and differentiate to produce active T helper cells, which will actually secrete cytokines to stimulate the mitosis of the B lymphocyte. So this process is called clonal expansion. Okay. So then they would undergo mitosis and differentiate into become B lymphocyte memory cells and plasma cells. So we know plasma cells secrete antibodies and yes, so then this will also produce memory cells. Same goes with this one. Okay. So lastly, the antibodies will help and destroy the pathogens. Okay. So coming back to this question, so they asked to describe the roles of B and T helper cells in establishing immune response against the antigen. So the answer is just what I explained just now. Okay. So the answer will be here. So yes, there is antigen in lymph and blood plasma. They attach to B antibody and B lymphocyte in the lymph node or the spleen. Then B cell will process modified antigen and present on cell membrane so that the presented antigen and self-antigen can be recognized by helper T cells. So that will cause the helper T cells to come to the area also to help. Okay, so yes, T helper cells produce a substance which stimulates mitosis and differentiation of B cells in activating them. Okay, so next question is suggests why activated B cell divides many times by mitosis to form clone population of cells. So quite simple, because they quite logic. Many cells produce so that yes, there's the bigger immune response, right? Okay. So you also have to say that all cells are actually identical so that they have to respond to the same antigen immune response. And next question, describe the role of plasma cells. So plasma cells, yes, they secrete antibodies against antigens, all right? You have to be specific. Okay, you have to mention that. So yes, and then you also have to mention the antibodies will be released to lymph and blood. So down here, you don't have to write also, never mind. It's just an extra info. So I also won't write on because I can't remember, but I'm not supposed to remember lah. Okay. So describe the role of B memory cells. 1 mark only. So B memory cells here. So I see helper cells here. They just retain the memory, okay, because they have the memory of like destroying the bacteria, okay, so that a quicker, more forceful response, so that they respond quicker because they remember. Okay, so distinguish between the primary immune response and secondary immune response. So as we see, the whole thing here will be the primary immune response. Here will be the secondary, okay. So primary immune response involves activation and multiplication of lymphocytes. We just explained the immune response, the one that we did here, but in briefly because it's only 3 marks. So this one also, just so you know, it takes 7 to 10 days to develop the proper antigen levels. Okay, so and then it also lasts about 2 weeks, so not long lah. Okay, so that's why the secondary immune response is longer. So because the memory cells live much longer, we have a better immunity towards these antigens. Okay, so but although it takes 2-3 days to develop, I mean, it takes shorter, yeah, it takes shorter time. Okay, so next question is about B lymphocytes respond to present non-self antigen as shown here. So here, this is a non-self antigen. So of course, they will elicit a immune response. Okay, so this is a B lymphocyte, so it starts to divide. Okay, divide. We call cells P and cells R. So explain what is meant by the term non-self antigen. Here. So yes, non-self antigen is definitely a macromolecule. It's a glycoprotein, or it can be, yes, a glycoprotein. All right. So yes, it stimulates immune response and is recognized by the immune system. Oh, sorry, that's just non-self. Yeah, correct. Okay, non-self. Huh? So outline how B lymphocytes recognize non-self antigens. Okay. Am I going a bit too fast? I'm sorry, I have to do this under 30 minutes. Okay, so outline how B lymphocytes recognize non-self antigens. So antibody, immunoglobulin, okay, antibodies bind to the antigen. Okay, and you have to mention that they are specific and complementary. Okay, otherwise, how else they're going to bind? Okay, so I think I can just skip this question. All right, so this one. Okay, so this one is cell-mediated response, all right? Cell-mediated response involving T cells. So here's the whole process lah. So the whole process, okay? So describe the role of macrophage in T cell response. So it's the same thing like the same thing that I explained just now, this one. So it begins with this, okay? So it's 4 marks. Okay, so here macrophages, they congregate, they come to the region of infections from chemotaxic attraction. Okay, then you know what they do, they engulf bacteria and digest them. Then they present on top of their cell membranes. They carry these antigens to the lymph node where most of the B and T lymphocytes are found, because you know they need help, right? So then they present the antigens to the T cells. Then they communicate by binding with the HLA receptors that in order to tell them lah. Okay, here the macrophage binding with this T lymphocyte. Okay, so because of that, the T lymphocytes also come and they activate the T lymphocytes, and then they come and help on the macrophages destroy the bacteria or pathogen at the infected area. Okay, so next question. So the sensitized T cells divide thousands of times to produce clone population active T cells. Suggest how this helps with immune response. So it's the same thing like this one, right? You're also asking like, why do we need a lot of clones of T lymphocytes? So the same thing like why also B lymphocytes divide. Okay, so anybody want to try and answer? It's just the same answer. It's the same answer as before. Okay, never mind lah. So yes, the same answer. So meaning the thousands of millions of cells, many cells available to destroy the antigen, okay? And then they are genetically identical. Okay, so describe the activity of killer T cells in the immune response. Killer T cells are involved in this one, okay? It's actually very simple, okay? So this guy, they're actually naturally born to kill. They're just like serial killers inside your body. So they attack 3 types of cells: infected cells, cancer cells, and cells with transplanted organ. So it basically kill your own cells also, okay? So yes, it all starts with the antigen presentation, because how else is the T lymphocyte going to know? So then they differentiate to T helper cell and killer T cells. So this is what the killer T cells, how they work, okay? They divide. Memory cells must be formed, okay, for these both processes. So then they will bind to the infected cell, all right, with the, they will bind to the infected cell. So the infected cell is also presenting the pathogen. Like when the pathogen takes over the cell, it also begins to make its own proteins, right? So the infected cell, in order to catch the attention of the T lymphocyte, it will take some of the proteins made by the cancer cells, because the cancer cells are using them. So they present it on top of their cell membranes also. So then when the T lymphocyte binds to this foreign substance on their cell surface membrane, it will initiate them to kill the infected cell. Okay, so yes, they punch holes, they secrete enzymes to punch holes, okay, so that they can put in toxins and destroy the cell completely. So once a cell is dead, the remaining parts of the dead cell will be eaten up by the macrophages. Okay, so they use H2O2 and free radicals to kill target cells. So describe activity of killer T cells is just the same thing like I said just now. Yes, so once they are attracted by the macrophages, all right, so the macrophages ask them to come and help. So then they just leave the lymph node and come to the site of infection to help them. Okay, so again, by chemotaxis, okay, because different kinds of chemicals are secreted that's just foreign and it's just very suspicious law for them. Okay, so then they collect around bacteria and secrete cytotoxic chemicals, they bind, then they inject chemicals, okay? So this one's also not in this one, secret chemicals, okay? But yeah, they call their gang to come also. They secrete the chemicals like lymphokines to call more of their gang to come and help, okay? Okay, what is the function of memory T cells in immune response? Same thing like just now. Does anybody want to try? Same thing like this one. Never mind lah. So same answer, okay. What is the function of helper T cells in immune response? So produce interleukin. Okay, they are the ones who produce all the cell signaling molecules to attract more cells, help them. Okay, so I'm having in the last part for my part antibodies. So yes, antibodies, yes, we can see they have two regions, antigen binding and the crystallizable site. This is the constant region and it's the variable region. Okay, so explain the functions part, label A, B, and C. So A is here, this is A. Okay, we now all know that it's the antigen binding site, okay? So they bind to the antigens, then they have similar shape. They're not complementary, they have similar shape, okay? So that's that. Then the B, part B is the disulfide bridge here at the hinge region. So this is the disulfide bond. You can't say hinge region because it points at the disulfide bond here. Hinge region is the whole thing. Okay, so they hold the polypeptide heavy chains together. Okay, so yes, it has a tertiary quaternary structure of the helps to maintain the shape of the antibody. Okay, then C will be constant region down here. Constant region down here. Okay, so they also, this is only to bind on phagocytes and macrophages, all right, not the antigens. Okay, all this one mark only, so just give either one. Okay, so last question from my part. Okay, so B lymphocytes have antibodies on the external surface. When B lymphocytes become plasma cells, they secrete antibodies. So figure 5.1 shows the enzyme papain. Okay, papain digests an antibody to obtain 3 fragments. Okay, so can anyone guess what papain is actually? Papain is an enzyme. It's actually found in, papain is actually the enzyme in papaya, you know, the papaya. Okay, so papain is found in papaya. Okay, so this is where, here the diagram shows where papain will work on, okay? Because papain cleavage site, meaning it works on here. Okay, so they break down from 1 to 3 fragments. Okay, so state 1 row of this region in the intact antibody molecule, meaning before it's broken down. So 1 row of this region. So they want the region where papain acts. So is this region, okay? So this region is called the hinge region. I think everybody knows this. Why is that a hinge region? So that it's very flexible. It's flexible, okay? So you can bind to differently spaced antigens, all right? So that's the function. Okay, the function of this main part here, only 1 mark. So that one also enough. You can go through the other mark scheme if you want. Okay, so state the function of 3 fragments, A, B, and C. So A, B is both the same because they're broken down from same, they're identical, remember? So we know that they are the antigen binding sites. Okay, they bind to the same type of antigen because they're the same but different, same polypeptide sequence as each other, but different from the fragment C. Okay, fragment C is constant region. Okay, so they bind to the monocyte, macrophage and neutrophils and B lymphocytes. Okay, so I think that's it for my part. That's it for my part. Sorry, I have to rush a bit. Okay, Philbert, I'm just going to stop share screen now. Okay.
On the steps of the production of monoclonal antibodies. So it asks you what happens during an immune response during step 2, where the mouse is left for a few weeks to allow an immune response to occur. This one already mentioned by Sharmaine a few times already, so I'm going to go through it very quickly. Antigen presenting cells by the macrophages so that the antigen can be recognized by the B lymphocytes. And then with the B lymphocytes with the appropriate receptors or antibody will undergo clonal selection and clonal expansion. Or you can say mitosis or cell division. You also can say T helper cells stimulate the B lymphocytes to actually mature into plasma cells. The T helper cells release the cytokines to stimulate the B cells to mature into plasma cells. Then the plasma cells will secrete the anti-A antibody because it's inject with antigen A. So this one, the next question, what is meant by a hybridoma cell? So can someone in the chat tell me what is the meaning of hybridoma cell? Should be very easy. If no, then I will just, okay, not enough time. Okay. Fusion of the myeloma and B cell. Yeah, but to be exact, is the plasma cell fused with the myeloma or cancerous or malignant cell. So why do hybridoma cells need to be formed? So because plasma cell can produce antibodies, but cannot divide, they are short-lived. While on the other hand, cancerous cell cannot produce antibodies, but can divide indefinitely. So you can, any valid point, you can, for example, you can say to obtain the genetic material from both cells. So how hybridoma cells producing anti-A antibody can be identified? You use a marker. Use the marker described, which is the antigen A that is injected in the mouse. So if the antibody can bind to this antigen A, agglutination or precipitation occurs, you can see it, then you can identify which one is anti-A antibody. So this one is talking about the RA, the rheumatoid arthritis disease. So it secretes this protein called TNF alpha. So monoclonal antibody infliximab is used to treat this. It will bind to this protein, TNF alpha. So here are 5 groups of people. These 5 groups of people each have different treatment. MTX is mentioned here is an anti-inflammatory drug. So at the end of the year's treatment, so they plot a graph to show you the results. These are the number of people in each group in the brackets here. So here it says describe the effect of the infliximab treatment on these people. First, you give the general idea that because if you see this one, group P is you only use the anti-inflammatory drug. You only can reduce the inflammation. It cannot actually treat the disease directly. So that's why the percentage is quite high, around 30.5%. But once you use the treatment, all these 4 groups also got used this monoclonal antibody treatment. They actually reduce by a great amount. So you can say that all infliximab treatments reduce percentage with increased joint damage. You also can give a general trend. You can say that these 2, group S and T, they use a high dosage. The high dosage actually have a lower percentage compared to the low dosage, which is Q and R, which has a slightly higher percentage compared to the high dosage. then both increasing dosage and decreasing time intervals have an effect. You can further describe by 4 and 5. You can say that at high dosage, so group S is 8 weeks, and then group T is 4 weeks. You can see that you double the time interval, but the change is only about like 0.5%. So you can say that the percentage is similar. But on the other hand, the low dosage, you double the time interval, you actually decrease by a good amount, like about 5%. So you can say that at low dosage, but increasing time interval, the percentage will reduce. And then lastly, you give like specific numbers. For example, without using the monoclonal antibody treatment, it's actually quite high, 30.5. But by using high dosage, you decrease until 0.5 to 1%, about 30% decrease. You also can give other comparative data. So suggest why the results in groups Q and R do not follow the general trend. The most logical reason would be the small numbers are involved, only about 60 to 70 people per group, so the data may be not so accurate.
Can you show the treatment? The treatment, because I can't get what the treatment is all about.
Oh, okay.
So S and T got higher dosage. Oh yeah, high versus low.
Yeah, and then
8 and 4 weeks.
8 and 4 weeks.
So 4 weeks is closer lah.
So no, the high dosage for 8 and 4 weeks is closer. This is the high dosage. S is 8 weeks, T is 4 weeks.
Okay, get it. Can you show again the suggest question, the last question?
This one?
Suggest the advantage of using monoclonal antibodies compared, no, not this one. Because a small number, just not the one. Previous one.
This one.
Suggest why the results in groups Q and R do not follow general trend. What is general trend? Huh?
The general trend.
Q and R do not follow general trend. What's general trend?
The general trend would be because if you see for the high dosage one, the time interval 8 weeks one will be higher than the 4 weeks one. But
that's the meaning of general trend.
Maybe I think so. If based on
you need to guess the meaning of general trend, no? Else I won't understand what is general trend. Suggest why the result in group Q and R do not follow general trend. Oh my God, what does it mean? Imagine you get this question in exam. I won't understand the meaning of general trend.
General pattern.
Unless the statement before this figure already state the meaning of general trend. Got not? Don't have, right?
I don't think
you need to figure out the meaning of general trend. My God. Gosh.
Okay.
Yep. Thanks.
The next question is explain the advantages of use of monoclonal antibodies compared with conventional methods in the diagnosis of disease. You need to take note that it's talking about diagnosis, not the treatment. When they talk about conventional methods, I also think need to guess because based on the marking scheme, they talking about 2 methods. One is microscopy. That one I think we all know, where you take a small sample and then see in a microscope. But the rest of the marking scheme is actually talking about culturing the pathogen, because different pathogens have their different living requirements, different growing requirements, because they prefer certain living condition or environments. So you can base on their growing characteristics to determine the type of pathogen. So if compared to monoclonal antibodies, you can say it's way quicker. So since it's way quicker, you then only can mention that you can treat the disease quicker. You can say that compared to culturing, it's less labor intensive because you need to actually grow the pathogen, and then not all pathogens can be cultured. For microscopy, you can say maybe the microscopic identification can be rather difficult. Viruses may be too small to be identified as well. And then any other valid point?
Yeah, can I add to this? So in the last time, when we don't have this monoclonal antibody, how do we identify the disease? Remember the nasal swab and throat swab? So we will be doing nasal swab, throat swab, or maybe you'll get your stool sample, your sputum, all right? And then we will culture the microorganism on agar plate. So just like how you do the antibiotic sensitivity test, remember? So you will transfer the bacteria that's in the cotton bud after nasal swab and throat swab and spread it out on the surface of the agar plate and put it into oven at 37 degrees Celsius overnight, 24 hours, to allow the bacteria to divide into clones. So when it's in the form of clones, we are able to see. So different kind of bacteria and species, the clone structure, size, color will all be different. So they will identify bacteria based on this clone of a pattern, structure, texture, and so on. So that takes at least 24 hours to grow. And some bacteria that are anaerobic, it will need about 2 days to grow them, okay, because they are very fastidious. It's very difficult to grow them. So that means for us to know what kind of microorganism infect your body, it will take at least 1 day or else 2 days. So it's too slow already. So therefore, now we've got this MAB, which can directly detect the presence of, for example, HIV antigen. Immediately within 5 minutes, within 10 minutes, we know whether this person's infected or not. So we can give a proper treatment, right? So therefore, this is faster, quicker diagnosis than to use the traditional method of culturing the pathogen. Okay, and then different pathogens need different living conditions and different kind of, some pathogens need blood to grow. So we need to grow them in blood agar, right? That means the agar is added with blood. Of course, not human blood lah. Okay, we're going to take horse blood or whatever blood. Okay, so that's the culturing of bacteria versus diagnosis using MAB is faster. Of course, microscopic examination also is like, not everybody can do that because you need to know how to identify the bacteria and you need to have a microscope and stain and so on. But for us to do it at home like HIV test, now we can do it because we've got a test kit, right? So MAB is very convenient and very fast and no need professional knowledge. So it's cheaper. Thank you. Pass to you.
Yeah, thank you. I try to faster. Okay, so this one is we'll be talking about types of immunization. So they're trying to study to find out the effectiveness of 2 types of immunization. So person A is receive an injection of antibodies against tetanus, while the other one received the tetanus vaccine. So these 2 graphs, this is person A, it show the antibody concentration against the time. So this is person B. So name the types of immunity shown by A and B. This one already give you a hint up here. It already say injection of antibodies and tetanus vaccination. So just straight away show the answer. So injection of antibodies will be passive artificial, and then the vaccination will be active artificial. Here say that if artificial omitted score 1 mark, if passive and active are correct. So if passive and active are both correct, but you didn't write artificial, they will still give 1 mark. So yeah. Next is saying for the graph, explain why the antibody concentration in person A decreased during the study period. So yeah. So it's saying mark 1 and 2 together, meaning you count 3 marks based on this entire question. So 1, you can say that the antibody is destroyed or broken down, or you can say the antibody is secreted. So why the antibody concentration did not increase? Because the immune response did not trigger. It inject antibodies, not antigen. So there are no active B cells or plasma cells or no antibody made. And any valid point, for example, further detail, lack of immune response, don't have stimulation for B cells by T helper cells, and no clonal selection and expansion.
Okay, I want to bring back to point 2. Antibody excreted means removed from the body. Removed from the body. So after phagocytosis, right? Broken down and therefore removed from the body, excreted.
So next one, it asks you to sketch on the graph B. So what happens that it inject a second booster vaccination after day 60. So hold on. Okay. Like something like that lah. Sorry, hold on. Okay, so your line must be increased, the antibody must increase at a faster rate than the primary response, and then must be at a higher concentration because this is the secondary immune response. Okay, explain why in this investigation, the experimenters measure the concentration antibodies of tetanus rather than the concentration of all antibodies. You will say that the antibody is specific for tetanus. You can give further detail about the variable region, which is the antigen binding site. You can also say that there will be always some other antibody molecules that is circulating